We're going to move on to the next type of biomacromolecule. Okay, we've already covered carbohydrates and proteins, two very different structures. But there's something that's missing in the picture we've built up for ourselves so far. We know that carbohydrates are a good energy source, and we know that they are also occasionally used as a structural thing. For example, chitin or lignin are used to make, for example, the exoskeleton of insects or wood. So we have this whole set of polysaccharide fibers. At the same time, we also have other kinds of structures, more active moving structures like muscle fibers and the like, that are often made out of proteins. We've seen that catalytic work, enzymatic work, is typically carried out by proteins, and proteins are basically all that there is to life. The vitality of life, the way things move, the way things get done, is all because of enzymes acting on all these other things, selectively performing certain reactions. So we've seen how these are all at the center, but we haven't yet looked at what the directing intelligence, in quotes, is. What is it that makes life capable of following certain goals, right? Because when we talk about biomolecules, no matter whether we want to study it as a subject as it is in its natural self, as in whether we want to study chemistry as it happens in a biological system, or whether we want to study how we can harness that chemistry in an artificial system, you have to always start off by understanding why each thing is there in the biological system, be that your inspiration or your subject. So a biological system has certain key tendencies, right? It tries to survive. It actively tries, right? A desk or a piece of chalk like this has no intelligence, has no need to try and keep itself alive, to preserve life. But most living organisms, except those that are trying to be a martyr or commit suicide, which does happen, like we have bees that enter martyrdom for the sake of the hive, what ends up happening is we always have this inbuilt self-preservation instinct, and to achieve that we have a whole bunch of other instincts, and in higher organisms cases, intellect. We prioritize things, we decide how to do them, and all of this we can do, with the brain at the biological level. But what is going on at the cellular level? What is the directing intelligence? So the directing intelligence is essentially found in something known as a nucleic acid. Nucleic acids, and they are called nucleic acids because they are found in the nucleus of the cell, which you guys will have studied since childhood. You will have heard the words, the nucleus is the brain of the cell. It's there in all those various mnemonic songs we had to learn. When we were in school, we had this rap song in 6th grade called Cells. Cells are made of organelles. If you've used that to study, you'll be familiar with this as well. So nucleic acids are so called because they are found in the nucleus of a cell. Now, what is it that they do? Unlike a brain in a higher organism, they typically don't have this adaptive response, right? Like where you think something and you say, okay, this is the situation, let me do this. These function through purely chemical means. So what happens is they are a code. They interact, basically the way different groups are organized on this nucleic acid is a code. So the first topic we have is the nucleic acids form a genetic code, and they carry information. And what does this mean? This means that there are particular molecules, you guessed it, enzymes in the cell that are going to interact with this DNA, and based on the arrangement of certain groups on it, okay, they're going to produce certain other kinds of proteins ultimately. So they're going to interact with the nucleic acids. I said DNA earlier, I shouldn't have. The nucleic acid can be DNA or RNA. We'll get into which one does what slightly later. But these nucleic acids, they basically interact with other molecular machines, with enzymes, and convey what the content is, just via chemical reactions. Literally, that's it. As the enzyme interacts with certain groups on the nucleic acid, it carries out certain functions. And what ends up happening is that ultimately a new protein is created. Why does that have to do with anything? Let's assume there's an organism that needs a particular protein to survive the winter. Say this protein is a peptide hormone that triggers hibernation. It's going to make the organism fall into a deep sleep and stop its metabolysis. Okay, so when there's cold, it cells sense the cold. Okay, and they trigger some receptor that makes the enzyme go and bind to that part of the DNA which has this gene. Now that enzyme will read the gene and create the protein ultimately. I'm simplifying now. We'll get into the actual process in a little while. And through a process, through processes called transcription and translation, it will create the proteins that are required to put the organism to sleep. Every action that takes place, every single thing we do, is mediated by switching on and switching off genes. by going and reading certain segments of the nucleic acid and retrieving the information required to construct corresponding proteins. And those proteins in turn can be enzymes that will go and assemble fats and carbohydrates into cells. Those proteins can be things like insulin that will go and trigger other cells to read other genes and then start figuring out whether to convert glucose into glycogen or fat. So there's this whole cascading process that goes on where proteins may be doing all the work, but nucleic acids have all the data, they have all the information, right? This makes them very interesting to study. Today, circuitry is fast approaching the smallest limit it can reach, right? Your transistors cannot get that very much smaller. Everyone's trying to look at alternate methods. People are talking about switching away from silicon to graphene, etc. But DNA and RNA are the most compact ways of storing data, and there are computer scientists and other scientists who are looking at using nucleic acids as a storage of data. So these could well transform, they've already transformed everything by giving us to life. They could transform everything we know a second time around, probably in a smaller way, by becoming the next great data storage facility. Okay, so what is the code of DNA like? There are 4 letters in this code. Okay, what does that mean? That means like just when you have a binary code, you have just 2 basic digits, zeros and ones, with which you carry all the information. And English, on the other hand, has several letters in the code, 26 letters, punctuation marks, etc. DNA is a 4-letter code, and we use the word letter advisedly. We basically mean like a character or a symbol. But what would have, and these are represented by A, C, T or U, depending upon which nucleic acid it is. T for DNA, U for RNA. So A, C, T or U, and G. These are the 4 letters, the 4 characters that we find in a nucleic acid sequence. But what is it that represents these characters? The way, as you know, in writing, we have different symbols for the characters. When we speak, we use different syllables, different sounds. And in binary code, we have different levels of current, right? You'll have studied this in computer science if you're a computer student, and maybe even if not, in lower grades. And you'll have studied it in physics in the chapter on electronics. So what is it that DNA uses? So essentially, each of these letters is represented by 4 aromatic groups. So there are 4 groups, 4 aromatic groups that are found along this chain. Why are they aromatic? Obviously, they're extremely stable. So you don't want too much change happening. You've already heard about mutations, I'm sure, which is how life evolves. When something in the DNA code gets messed up, gets changed, and then a new protein is created, and a new kind of organism begins to form. So you don't want that happening all the time, because then like nothing would be the same. All our proteins would change completely. So these are nice stable groups, 4 aromatic groups. Okay, and these have been ligated to a long copolymer. So what is a copolymer? A copolymer is essentially a polymer with different kinds of basic residues, different kinds of, how should I put this? Fundamental units that are repeating, different kinds of repeating units. And the whole just like lactose was a dimer of 2 different monomers, right? They weren't identical. So if you had that repeating and took a polymer of lactose, that would be a copolymer. So here, there's a long copolymer with 2 different monomers. So you can see I've done them in green and blue, green and white, green and white, green, white, green, white. And at the end of every green one, we have these aromatic groups ligated onto it. Maybe I should have put them over here. These are ligated to the green, not to the white. I've put them at the juncture. But anyways, now the most important thing that you need to know over here is that though it's a long molecule, right? And we know now from our study of polysaccharides and proteins that long molecules get highly folded, right? We've seen the left-handed helical structure of amylose, the right-handed helical structure of alpha helices. We've seen the sheet-like structures of cellulose and beta-carotene, say. So now what we're talking about is that even DNA will exhibit secondary and tertiary structure. But the information we're interested in, the info, is encoded in the primary structure. It's encoded only in the sequence of molecules, of atoms. It's not encoded in anything else. It's not encoded in it's not encoded in the folding. It's not encoded in your structure. Whereas for proteins, right, often the most important thing they do, their functional importance, is based on their structure. For these guys, for nucleic acid, it has to do with the direct sequence of your basic nucleotides. It has nothing to do with the overall structure, the information that's been encoded. The information that's been encoded is useful because it can help it structure it has is useful because it can help it interact with enzymes in certain ways, maybe. to bind to them, but when the information is being read, it's always the primary sequence only. So we'll spend quite a little bit of time on primary sequences. Okay, so what we're going to do now is we're going to first look at the structure of this long copolymer. This long copolymer, we're going to look at what its structure is like. Then we're going to move to, so we'll say, okay, once we know the structure, we're going to move to the structure of these 4 aromatic groups. We're going to talk about how those contain information. Then we're going to analyze the secondary structure of DNA, and we have a nice story to tell in the meantime about its discovery and photograph 51, which has been the subject of a play. And and then we'll move on to how exactly the code is read. So the basic copolymer that the nucleic acids are founded upon is a very sweet and sour sort of molecule, literally, because they're made out of one sugar and one acid. So there's like acidity in it, and there's also a carbohydrate in it. So we know that there are 2 different nucleic acids we talk about often. There's RNA and there's DNA. So the reason there are 2 different nucleic acids, like what is the main chemical difference? The main chemical difference is that there are 2 different basic sugars at the base stuff. So I want you to try and name this sugar. So you can pause the video, take your time. Sugar nomenclature is difficult. Take your time and see if you can name this sugar. Okay, I hope you've done that. So let's see how you might go about naming this structure, right? So first thing to see is that it is a furanose. Clearly, 5 of them, so a furanose. So it's a furanose. Now a furanose has 1, 2, 3, 4, 5 vertices, and you can also see this has 5 carbons, right? 1, 2, 3, 4, and the 5th outside the ring. So it's a pentose of some sort. Is it an aldopentose or a ketopentose? It is an aldopentose. How can we tell it's an aldopentose? Well, this oxygen is clearly the hemiacetylic oxygen. So which carbon, on which of the sides, is there going to be a carbon that bore the double bond O before hemiacetylization? Well, this carbon has got an OH, which the hemiacetylic carbon will have. After hemiacetylization, the same carbon will bear both the hemiacetylic oxygen and the OH. This carbon doesn't have an OH. It has a CH2OH over here. So obviously this was the acetylic carbon, and this was a carbon that just had an OH before that has participated in the hemiacetylization. So if this was the hemiacetylic carbon, you can see it has only got another 1 carbon attached to it. It doesn't have 2 carbons attached to it. So this must be an aldopentose, right? Now what are the aldopentoses we have? We have ribose, arabinose, lysose, and xylose. All of them divided into D and L. What is the configuration over here? Well, we can see whatever is on the top must have been on the left, whatever is on the bottom must have been on the right, except in this position where there is an inversion due to the double switch. We can see that this guy is on the top and the last position. So if we perform that inversion that we're supposed to perform, then the H would have been on top, not on the bottom. And if the H was on the top, that means the H was on the left in the Fisher projection. If the H is on the left in the Fisher projection, then OH is on the right in the Fisher projection. So this is a D sugar. So it's D something, okay? It's a D sugar. Now what is the basic sugar over here? Now I just told you ribose, arabinose, lysose, xylose in that order with respect to whether they are alternating or identical OH groups. OH down, and back here also initially OH down, OH on the right hand side. When all are the same, it's ribose. So D ribofuranose. But we're not done yet. What about the hemiacetylic group carbon? Is it which anomer is it, alpha or beta? Well, since this is D-ribose, before the hemiacetylization process took place, the OH must have been down. And since this is in the opposite direction, this would be a beta D-ribofuranose. And that is what we're interested in. Beta D-ribofuranose is the basis of ribonucleic acid, RNA. What about this? You can see the structure is the same. O, OH, OH, H, 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 OH, H. So you can see here that there's a mismatch, right? So this is therefore known as 2-deoxy beta D ribofuranose. Two deoxy beta D ribofuranose. And this is the basis of DNA. So this gives rise to deoxyribonucleic acid, this gives rise to ribonucleic acid. And DNA is known as deoxyribonucleic acid because it's made out of a deoxygenated version. You can see over here that this is deoxygenated. And that's because if you have this OH over here, it actually makes it more unstable in the long run. When you have this long polymer with this sugar constantly recurring, then that second group, due to the chemistry of sugars, which in reactions that we haven't had the time to delve into, there are reactions that can occur where having this OH over here makes it possible to break the ring with different groups coming and attacking. This 2 OH being missing makes DNA far stabler, which is why DNA can occur for long chains, whereas RNA is limited to much shorter chains. We'll get back to this. Okay, but having said that, how are these different groups linked? So I just told you that it's a copolymer, right? So there must be this other polymer, this other monomer we need to look at. If this is the sweet, the sugar, then the other one is the sour, the acid. So it's called an acid, and this is the reason we have nucleic acids. This should be a very familiar acid to you. This is nothing but orthophosphoric acid. They studied it in P block, big deal, nothing much to talk about over here. Except that you should know that this group is very popular with biomolecules. But I don't mean in biomolecules, I mean with biomolecules. Like biomolecules have an affinity towards this. Life has evolved to use this in almost every possible situation because, as we covered at the very beginning of the class, this guy can make very strong bonds. It makes very strong bonds. And so when you have molecules like ATP or ADP that contain these phosphate groups, they're released and they go and bond onto something, and the formation of that strong bond releases a lot of energy. Conversely, that also makes it very useful when you want to create this long stable structure. So let's look for a moment at how these fit in with these to create a long polymer. So let's take the case of a simple ribose. So here we should have a CH2OH, right? Now, if I draw this in its neutral state instead of in the ionized state, okay, this is to help you understand the reaction. It can react in either state. What's going to happen is this OH over here will react with this OH over here, and a water moiety will disappear. This H will come away. It will become, like I showed you before, minus and H plus. H plus will come and attack over here. This will become OH2 plus. This group will be removed. The positive charge will shift here, and then this guy will come and attack over here. So the ultimate group is going to be this. A P in a circle is the symbol often used for a phosphate group, just to indicate this whole group rather than write the whole thing again. Okay? But this doesn't make a copolymer, this makes a dimer. This needs to be able to keep polymerizing, right? So it has to, if it polymerizes in one direction, it has to be able to polymerize in the other direction. So just like this, another one of these phosphate groups will come and attack over here. I'll just draw it in full this time to indicate how it's working. So as you can see, what's ended up happening is this guy has polymerized like this. It's bonded to a phosphate group, which has in turn bonded to the fifth carbon of the next ribose, which has bonded to another phosphate group here, which will via its third carbon, which will bind to the fifth carbon of another one. So you can actually, if you imagine this P and this P to be the same phosphate group, you will see how you could make a cyclic dimer. Cyclic dimers generally don't take place. What actually happens is that this is a long chain running on forever. This end is called the 5' end with a dash like that if it is unpolymerized. So The last one that isn't added onto it, that doesn't have any more p's attached to it, is labeled as 5 prime. And the last one here, if I go on further where there's no p attached, finally, is the 3 prime end. We'll come to why this prime symbol is used in an instant, really. But this is the basic understanding that we need to have. These two monomers are co-polymerizing and they repeat on ad infinitum. And that is the basic structure on top of which the nucleic bases are going to be added. They're going to be ligated onto this. So you should understand the structure very well. Also, you might get a question. You might get any of these questions actually. You might be asked what links carbohydrates. You know the answer is the glycosidic bond. If you look at what links proteins, the answer is the peptide bond. And here, this linkage via phosphate group is known as the phosphodiester bond. Phosphodiester. What does that mean? Okay, so normally what we call an ester isn't just an ester, okay? That what we call an ester is short for carboxylic ester. The product formed when a carboxylic acid attaches to an alcohol, right? This is not that. This is a phosphoester. When the product formed when a phosphoric acid binds with an alcohol. Cool? And in particular, since the phosphoric acid has got 3 OH groups or 3 O minus groups, depending at when you catch them in their reactivity process, it's actually a phosphodiester, esterifying on both ends. The reason that a nucleic acid is a nucleic acid is because this third O is left as an O minus and it does release a proton into the solution. So your DNA actually has a huge negative charge. How is it counteracted? Remember in our discussion of quaternary structure, I had mentioned a protein called the histone protein, which is it's actually an octamer. It's made out of 8 different proteins and it binds DNA and helps it wrap up compactly, right? That histone octamer is positively charged and that is what makes up for this. Otherwise, your DNA would be filled with such a negative charge, it would never be able to coalesce. The whole thing would, erupt outwards, and it's that fragment, that fiber would go back and forth to the sun thousands upon thousands of times. That's the amount of DNA that's stored inside your self. We'll discuss it again when we come to the precise secondary structure. Anyway, so that's the difference between, to wrap this up, RNA is based on ribose, DNA is based on 2-deoxyribose. The reason for this difference is that 2-deoxyribose is less susceptible to certain reactions that can cause ring breaking, therefore it is more stable. These are polymerized with a phosphate group, which is a tribasic acid based on phosphoric acid. This phosphoric group forms a phosphoester with the 5th carbon, which is noted as the 5 prime end when it's free. But once it binds on, it is no longer the 5 prime end. This phosphate group comes here, and whichever ribose it bonds to above that will have the free 5 prime end, the one with the OH group. And at the bottom here, there's the P over here, and if instead of the P we had an OH over here, that would be called the 3 prime end. So there's a 5 prime end and a 3 prime end, and they're polymerized via phosphodiester bonds. And that brings us to a conclusion to the structure of the long copolymer. And now let's get into how the information is encoded in our DNA and in our RNA. Let's look at the actual coding aspect. We're going to come now to the first type of nucleic base. Yes, you heard that right. I said they're aromatic, but there are 2 different kinds of aromatic ring-like structures that these nucleic bases can employ. So we're going to basically what I'm saying is that we talked about 5 nucleic bases, right? A, G, C, T/U, because T and U occur in distribution. Either you have T in DNA or you have U in RNA. So these 5 bases, okay, they come into 2 classes of compounds. 3 of them, U, T and C, come under what we know as the pyrimidines. Okay, and the remaining 2, A and G, come under what we know as the purines. So we're going to study the pyrimidines first, and then we're going to study the purines. Okay, we're going to look at their structure. So what is their structure? This is pyrimidine itself, without any substitutions, any modifications. Okay, so since I should probably write this in the plural, since we're looking at the substituted versions also. So simple pyrimidine is very similar to pyridine, which you must be familiar with. But here, instead of substituting 1 carbon as a nitrogen, we've substituted two carbons with nitrogens. Okay, and the substitution is 1,3. They're not adjacent, they have one in the middle. 1,3 substituted. Understand, when I say 1,3 substituted, I don't here mean that there's a meta substitution going on. I'm not substituting the hydrogens on the carbons, I'm substituting the carbons themselves. This is a heteroatomic ring. And both these lone pairs are active, they are present in the reaction. Because they can be used to be basic, because this resonance is already complete without them. So these pi electrons will not be parallel to the other to these p orbitals, it will be in some other plane. Cool. Now, the first modification we do is common to all the three pyrimidine bases. What is it? We make this bond. You can see this bond here, right? We're going to break this bond. We're going to make it a double bond O. And we're going to introduce an H over here. Now, how does this work? Well, obviously this is resonant, right? So we can always make this happen. So look at this structure. When this bond comes here, if I make this a double bond O, then that bond with this nitrogen, the lowest nitrogen, is lost. So the lower nitrogen now has a free bond, and that goes and binds to this H that we've added. Okay, and this bond has come over here, this bond has come over here. So basically the reaction took place with the other resonance structure, not with this resonance structure, and we got this kind of setup. Now, why is this common to all of them? This is essential because it is through this NH that we will bind to our ribose or our deoxyribose sugar. Remember, in the deoxyribose sugar, we had a structure, right, where we had on this first carbon, we had an OH. So this OH and this NH will go off as H2O, and so there will ultimately be a straight bond from this ribose to this end. So unless you have that H over there initially, you're not going to be able to make that bond. So you can see that all three of these structures, have NH over here and this double bond O on this side. Okay, but equally well, we can drop this H and write it as a free bond, because ultimately it's going to just add on to the ribose or the deoxyribose sugar. Okay, depending on which one it is. So now let's look at the further modification. We need to convert this into three different compounds, right? So we're going to have to break up some more of these double bonds and add some more groups. So one thing you can do is you can add another double bond O over here. That gets you this compound. Okay, I don't know if any of you know what it is. It is uracil. So let's write that down somewhere. Let's change color for it. Uracil. And the thing about uracil is that it has both these double bond O's over here, and therefore the resonance in the ring has kind of been lost. What remains is basically this one group. I mean, yeah, there is still some, you can resonate like this, but they're not equivalent structures anymore, which they were earlier. This has made the ring somewhat more unstable and has made this final remaining double bond fairly vulnerable to attack, and therefore it can be mutated, and we want DNA to be stable. So while this uracil or U is often found in RNA, it is one of the 4 bases in RNA. When we come to DNA, we methylate this. We add a methyl group here without disrupting the double bond. Remember, the double bond is not disrupted. When this methyl group is added, we get what is called thymine. So uracil and thymine is what is found in DNA. So U in RNA, T in DNA, U in RNA, T in DNA, with this being methylated at a position. So if you're asked what is the difference between uracil and thymine, the answer is thymine is methylated uracil. Okay, thymine is methylated uracil. So to wrap back here, we made the first modification, which is breaking open this double bond here. So we got double bond over here, that is common. And then from this we had an H, which can also be replaced by a bond leading to whatever the sugar is, ribose or deoxyribose. For this one it must be deoxyribose because it's only used in DNA. This one it must be ribose, it's only used in RNA. What about this guy? This guy is the other one of these three. It is cytosine. Cytosine, also written C, just like this is U and this is T. Okay, so cytosine is also written C, and what it is something similar to this, but the second modification, instead of being a double bond O, is the addition of an NH2, and this bond is maintained here, and therefore there is no need to methylate this and preserve it. So there's only one version used for both DNA and RNA. Okay, this NH2 being a single bond just replaces the hydrogen that is present here, which obviously we're not marking out because the bond line structure. Hydrogens are deemed to be implicit. So we don't need to worry, whereas in this case we have introduced a double bond, and so we had to break one of these bonds already, and therefore make this last remaining bond more vulnerable. Here these two are equally vulnerable, so there's been an NH2 added in, and obviously this NH2, if you draw out the H's, this is capable of donating a hydrogen bond. We must never forget that this is capable of donating a hydrogen bond. Okay, so cysteine will bond to guanine, right? And it's going to take, this guy is going to donate some bonds, guanine is going to give it some bonds. We'll come to that in a bit. I'm getting ahead of myself. But just keep track of the kind of groups we're associating here. Okay, we're adding double bond O's, we're adding N's with hydrogens, and this N is staying over here. Okay, just keep track of what these groups are like, because when we come to the second structure of DNA, the specific substituents we've added will be very important. Okay, and there'll be some specific conclusions we'll make from that. Anyway, these are the three pyrimidines. Now we move on to the purines. So now we're going to be talking about purines. Purines are the other major kind of nucleic base, and unlike a pyrimidine, well, actually similar to a pyrimidine, they have this structure with two nitrogens substituted into a benzene, but this is fused on with another ring, which is a 5-membered ring, again with two nitrogens. The cool thing here is this ring, actually, these two rings actually share this double bond, and it is part of both of the resonances and makes both of them aromatic. But the lower nitrogen of the 5-membered ring does not have a double bond on it, and so its lone pair of electrons is permanently involved in this delocalization. However, as a consequence of not having a double bond, this nitrogen has a free third valency. In its native, when the purine is by itself, this valency is borne by an H, but oftentimes it will be reacted and it will be borne by the deoxyribose or the ribose sugar at the first carbon. Okay. Now, this particular, as a result of this, as a result of the integrity of, the presence of this bond, the integrity of this bond over here, this double bond in pyrimidine, which was disrupted in all the pyrimidine derivatives, it will remain, like the integrity of this bond is not hampered or destroyed in any of these derivatives. You can see that this bond has remained in both these cases, right? Both the purine derivatives. Because it doesn't need a bond over here to bond onto the ribose or deoxyribose sugar. That is taken care of by this floating bond over here. So the question comes to, what are these two like and what are they called? Now, this guy over here is simply a modification by adding an NH2 group. This is a modification by adding a double bond O and an NH2 down here. And this double bond O has to break this bond because it needs a second bond, right? It's not just replacing the hydrogen. And that causes an H to appear here. So this is actually the most derived, the most number of substituents have been added on here. And this is called guanine. This, wait for it, this is a very familiar compound. This is adenine. If you remember, AMP, ADP and ATP were adenine monophosphate, adenine diphosphate and adenine triphosphate. So this is the basis of the energy currency of all the cells. Yes. So now we can move on without ado to the secondary structure of DNA, so that we'll understand how this polymer is folded up. We know it contains these 4 bases on it. Once we get to the secondary structure, we'll understand why these substituents were used, because they have a key role in organizing the secondary structure. And once we've got that, we can move on to how to read and decode this entire information. You might wonder why we need to study the secondary structure at all, if what we're interested in is understanding how this information is coded here. And the answer is that the enzymes that bind onto it will bind onto it in such a way that makes the secondary structure important to understand. It's relevant to the enzymes that bind onto it, which then carry out the reading of this data. Okay. So let's come to the secondary structure of DNA specifically. because RNA has a very complicated array of secondary structures that are not really in our syllabus, except one of them. So the fact about DNA is that the solving of DNA structure was a landmark moment, a watershed moment in the history of science. Soon after we solved its structure, we began to see how it was the perfect candidate for a molecule that could carry information about life. The molecule that could, in a short and elegant code, have everything that we need to know about how an organism has to survive. It was wrapped up away in the nucleus. It was a perfect candidate for almost everything we had to do. The puzzle of genetics. Because if you recall from class 10 biology, Gregor Mendel had come up with a theory of genetics back in the 1800s, right? With tall pea plants and short pea plants, and he interbred them and whatever. But that worked for some 7 traits, like some 7 different characteristics of pea plants. It didn't work for the vast majority of them because, as you now know, what actually happens is the genes get read and converted into proteins. But though Gregor Mendel's ideas were initially dismissed, over time it became apparent that there was something to them. Maybe not everything comes in this binary opposite, that either this protein is produced and then a trait is there, or the protein isn't produced and the trait isn't there. Some things were lurking behind it. Like people discovered that there was a combination of traits, combination of proteins that you could express, and if you could express all of these proteins, then you would get the trait. And if you couldn't express one of them, then your trait would change, and so forth. And people realized that all of these traits, these proteins you can produce or can't produce, must be encoded in something, right? And DNA became the leading candidate for what it could be around the same time as when we solved its structure. Because that gave us this very clear idea, okay, this very highly stable compound, very tightly wrapped up inside the nucleus, very long, so it can store a lot of data, might be the candidate. And soon after that was confirmed as well. So in the 1940s, there was a young man called E. Chargaff. Okay, he would have been in his 30s back then. And he stated this rule that is known as Chargaff's rule. The ratio of pyrimidines to purines, not pyramids. Sorry, my bad, not pyramids. Ratio of pyrimidines to purines in a DNA sample. Okay, again, we're talking about DNA over here. But this particular rule is also valid for RNA in a modified form. So ratio of pyrimidines to purines, that is the ratio of C plus T over A plus G, is equal to 1. So the same number of pyrimidines as there are purines. Or you can express it like this, C plus T over A plus G. Okay, there's a reason for this expression. It turns out that independently, this was not done by Chargaff, this was discovered later on. Independently, T by A is equal to 1 and C by G is also equal to 1. Okay, so if T and A are equal to some amount, say X, and C and G are equal to some other amount, say Y, because they have to be equal to the same thing, then this is basically X plus Y divided by X plus Y. If I replace C with G and I replace T with A in the upper expression, you can see just something divided by itself. So then it becomes no great mystery. But these ratios were not known to Chargaff at that time, nor did they know at that time why these ratios were correct. We'll get to that. Okay, so now there were X-ray studies that commenced at King's College. Now, what is an X-ray study? We've touched upon it briefly before. So there's a method called X-ray crystallography, where you crystallize something. You crystallize some compound, and then you shoot it with X-rays. Now, what happens here is that these X-rays will get diffracted. They will bend around each of the various atoms, and then they'll interfere with themselves, and they'll give us characteristic diffraction pattern. Okay, just like you do in physics when you have a diffraction pattern between 2 slits, right? So here, if you have 3 or 4 atoms, then the slits are the gaps between the atoms. Based on the diffraction pattern and the size of the diffraction pattern, you know that fringe width and the fringe distribution can be correlated to the size of the slit and to the distance between slits in diffraction and interference patterns. So this way we can correlate the diffraction pattern we get to the size of the individual atoms, and we will know where each atom is in a crystal. Okay, now one of these major scientists was called Rosalind Franklin. Okay, she is one of the most famous scientists of the last century, one of the most important biologists or rather chemists of the last century. She began with a PhD where she studied holes in coal. Literally. So you've studied so many reactions where you say charcoal is active or coal is active and it has adsorption. So she used these X-rays not to study crystalline substances, which are periodic and recurring and easy to study. She used them to study amorphous substances like the surface of coal and charcoal, and that makes the data very noisy. Can you imagine if you've got an X-ray of a crystal, then from every angle you're going to get a repetitive, recurring pattern. It's very easy to confirm that your data are right. But in amorphous, by definition, the pattern is never going to be the same. So it's very hard to decide approximately what the pattern is like, because the data are different from every angle. So we have no way of figuring out, when you come to multiple conclusions with doing an X-ray of a crystal, you can always say, okay, this conclusion is borne out by data sets taken from all angles, so it must be correct. The others are only borne out from one or two angles, so they're wrong. But in an amorphous case, all your data sets look different, so it's very hard to decide what the actual structure is. But she did that, and she studied the structure of coal. So then she joined this lab at King's College, and the research director there was very strict about how, they had very little personal autonomy. So she was paired with another scientist called Wilkins, but she was this very assertive and strong person. And this is the story I promised that you'll get about the discovery of DNA. Wilkins was a very shy fellow, and there was a clash between them. So ultimately there was this division in work. She was given a form of DNA. Where do I write this now? Filled up the board. So Franklin studied what is called aDNA. This has nothing to do with adenine. A for adenine is a particular thing, and A for aDNA is a particular form of DNA, particular secondary structure. They got bored of using alpha and beta for everything in biomolecules, so they began using Latin letters, Roman letters, or I suppose English letters if you want to call them that. They began using the Latin script, and Wilkins was given bDNA to study. So they split the project so they wouldn't have any more personality conflict. However, Wilkins's doctoral student, Raymond Gosling, was given to Franklin. So Franklin had this student called Raymond Gosling, and they worked for many years, and they did these brilliant studies. She was one of the most meticulous scientists of the era. So she would actually never predict anything until she had all the possible data. So she had immense patience. She would collect data at a point where other scientists would say, "Arey, this is clearly this. Let's just go ahead and make a prediction and say this is what the structure must be like. Publish it. Let the whole world know. If we're wrong, we're wrong. Someone prove us wrong 2 years later, but let's go get a promotion today." Franklin wasn't like that. She believed in getting absolutely 100% accurate results, which is not always the smartest move if you want a quick career advancement, but it is the most meticulous. And she and Raymond Gosling did this fabulous analysis. Now, around this time, Watson and Crick were two biologists, and they thought that DNA might be this candidate for the genetic material, and they were studying its properties and what its structure might be like based on whatever they knew. But they were biologists, remember. And they had come up with some models, and they weren't quite sure, and they tried to collaborate with this lab. Now, they record that Franklin was never cooperative, but if you read like Watson's autobiography, you can see that they never really respected Rosalind Franklin very much. You must remember, this is 1953 we're talking about, and at that point in time, there were very few women at the forefront of science like she was, and they probably had this kind of sexist misconception of her as a weak scientist or not a very smart woman. They discounted a lot of her predictions and so forth. And anyways, it was time for Rosalind Franklin to leave King's College and move to another institute. At this point in time, her PhD student, Raymond Gosling, was transferred back to work under Wilkins, who was studying bDNA. And this was the controversial part. Wilkins got Raymond Gosling to show Watson and Crick this famous photograph called Photograph 51. Photo 51, which was a photograph at the molecular level of what DNA looked like, based on the X-ray structures that the structure that they had elucidated via their X-ray approach, their X-ray photography, crystallography, and spectroscopy. So this result, Photograph 51, was startlingly clear, and they could see how these molecules were arranged. And Watson and Crick immediately, since they had no compunctions, they went and they drew where the hydrogen bond should be, and they made a complete prediction, and they turned out to be right. And Rosalind Franklin had not yet written a prediction about it, but she had written a paper describing this structure. And Wilkins never told her that he had shared this photograph beyond the lab. She wasn't even aware that Watson and Crick were working on it. She had no opportunity to contribute to the building of the model. Ultimately, Watson and Crick published their paper first, and they credited Wilkins mainly, and they did mention at the ending this was inspired by some work by Rosalind Franklin, and Rosalind Franklin and Wilkins' papers came out later on in the journal. So instead of it being Franklin finding this beautiful structure, which she did, and then later on these people using it to formulate what the bonding must be like, what ended up happening was Watson and Crick said, we have discovered this brilliant model, this is what DNA is like, and we were inspired by some as yet unpublished work by a woman called Rosalind Franklin and her colleague Wilkins, who helped us so very much. And afterwards, her paper came out afterwards, and as a result it looked like she was just backing up and providing support for Watson and Crick's model. This isn't to take away from what Watson and Crick did. Their ingenuity was crazy. The way they worked at it and the way they solved the structure, figure out where the actual bonding must be to make the molecule look like this, where all the interactions must be, that was also really great. And that also took a lot of effort. But it is quite true that there is some shadowy, like some sort of doubt and controversy about whether or not Rosalind Franklin was given anything as near as much prestige or respect as she warranted. Unfortunately, she died soon after at the age of 37 because of ovarian cancer. There's a theory that maybe working with the X-rays without protection like the kind we use nowadays may have caused the cancer. She died very young, and 1962 the Nobel Prize was announced and she was already dead, so they didn't bother nominating her for it. And as a result, it was given to Watson and Crick and also Wilkins, who shared the data without asking her. And Photograph 51 later became the title of a play that chronicled her life and the work, not her life, but her research and the work she had done to obtain this photograph. So that's a very interesting tangent. Anyway, coming back strictly to science, what was it that Watson and Crick came up with in this model of theirs? It was based off this brilliant structure that Rosalind Franklin had photographed with her PhD student. Well, the structure that they determined was a DS hybrid, we call it, a double-stranded hybrid. That meant that the secondary structure wasn't some kind, like when we studied for proteins, we studied sheets like this and helices like this. It was nothing of the sort. Rather, it was a DS helix, a double-stranded helix, something like this. Right? So you can see very clearly that both the strands of the helix were intertwined together. Okay? They were intertwined together. And how was this possible? How was it that these two strands intertwined together? It was possible because each adenine on one strand of DNA, say there's an adenine over here, would be pairing up with a thymine on this strand. How would it do this? Adenine would provide one hydrogen bond and receive one hydrogen bond from thymine. Okay, so wherever there's an adenine on this side, there will be a thymine on the opposite side. Wherever there's a guanine would provide two hydrogen bonds and accept one. Cytosine would accept two and provide one. So they were also naturally locked together. So there were also guanine-cytosine pairs. Guanine-cytosine pairs. And what ended up happening is that these are the gluing force, the sticking force that glues the two strands together, and that is why DNA tends to occur in a double helical form. RNA, on the other hand, tends to occur in a single helical form. There are exceptions. There exists single-stranded DNA, there exists double-stranded RNA. Both of them are primarily found only in viruses. They're not found in living organisms. Living organisms restrict themselves to double-stranded DNA and single-stranded RNA. And this double-stranded DNA, we'll come to why it's double-stranded. What is the biological function of this double-strandedness very quickly. But you must understand that it is supported by these hydrogen bonds. So let's now magnify this structure, which is the bDNA. It so happens that they worked on two different structures, aDNA and bDNA. We don't even have to worry about aDNA. It's not like alpha and beta where both the proteins were hyper important, because aDNA typically occurs when you dehydrate bDNA. They are interconvertible, and in our body, generally DNA is always very highly hydrated. It's surrounded by water, and so we'll only worry about the bDNA. We'll look at its structure and we'll look at how this hydrogen bonding is taking place inside it. So let's move on and do that. Right, so here is our gorgeous bDNA structure. I haven't drawn it very well or not much to scale. So I hope you get the gist of it. There are a few major things to note. Firstly, it's double helical, obviously. Secondly, there is a directionality to DNA, as I've already told you. In a nucleic acid, one end will have an OH left unbonded, without bonding to any phosphate group, without any esterification, at the fifth carbon, right? You have this long chain, like what's happening in a long chain like this. This is one ribose, then beneath that I have another ribose, and beneath that I have a third ribose. Then what's going to happen is, how are these going to be linked? There will always be a fifth carbon here that will link via phosphate group to the third carbon here. Fifth carbon here will link via phosphate group to the third carbon here, right? So what happens here? This guy is fifth carbon. It's free. It's not bonded to any phosphate group. It's just got an OH over there. That is called, so this direction towards the free fifth carbon is known as the 5' direction. And the opposite direction towards the free third carbon is known as the 3' direction. I've already mentioned this once, but I'm doing it again because it's rather important that you should understand the notion of this directionality. Because yet again, we're going to classify this helix as either right-handed or left-handed. Remember, to classify a helix or right-handed or left-handed, you have to look at the bottom, imagine it like a staircase, and go towards the top and see whether the staircase goes from right to left as it goes from bottom to top, or whether it goes from left to right as it goes from bottom to top. But to define bottom or top, we need a directionality, right? So you look at the 5' end and you go up. You take the 5' to be your bottom and the 3' to be your top. But you might say that doesn't make any sense. That you have two strands like this, one on either side. So if you do you look at the bottom of this strand, like the 5' of this strand, or the 5' of that strand? Luckily enough for us, is it, because let's just imagine for a moment that it is 5' at both. It's 5' for both strands at the bottom. Let's imagine that the bottom has 5' for both. Then you can't classify it as a right-handed or left-handed helix, right? Because as I go from 5' to 3', there'll be one helix going rightwards and one helix going leftwards. Luckily for us though, what happens is always that if one of the strands ends in a 5', opposite that the other strand will always end in a 3'. And similarly, if this strand, now this is here the orange strand was 5' here, so its 3' end is up here, and therefore the yellow strand has its 5' end over there. So now, if I look from here and I follow the red strand, because it's going up, because this is the bottom of the red strand is at 5', top of the red strand at 3'. Clearly, it's a right-handed helix. If I look over here and look at this as the bottom of the yellow strand, this 5', then again from that direction, it's a right-handed helix. So it's a right-handed helix whichever way you look. That's a key feature of B DNA. It's right-handed, and the directionality is from 5' to 3'. Opposite ends are together, are the same. Opposite ends of two strands are at the same end of the double strand, so to speak. So if the end of the orange strand is 5', then right next to it there'll be the 3', and vice versa. Okay. Next, what is the length of each turn of the helix? 34 angstroms, and that's approximately 10 base pairs. A base pair is basically one base on either side. As I've already told you, the reason these are stuck together is because the bases on either side are pairing via hydrogen bonds, right? That gives us and approximately 10 such pairs of bases will correspond to one turn. Okay, and the width of this is roughly 20 angstroms, but actually every alternate groove is wide or narrow. This is wider, this is narrower, this is wider, this is narrower. Okay, so 20 angstroms is sort of an average over there. Cool. Now let's look at how the actual bases are pairing up. So let's say this is a ribose sugar, okay, with the 3' end free, and this is the phosphate group. Okay, wait, I'll come to this later. Let's do the upper one first. The upper one I haven't included the ribose sugar. In the lower one I've included the ribose sugar only to explain why this weird thing happens of 5' and 3' going together. So let's come back to just the upper one for now. This is, I hope you can pause the video if you like and identify which these two are. Okay, I hope you did that. This is guanine. This is cytosine. Now, let's look at what happens in guanine and cytosine. Guanine, as you know, it has this double bond O introduced here, and because of that, it had to break this bond. One second, sorry. Yeah, it had, since the double bond O was introduced, this double bond here had to be broken, and so this N has now acquired a hydrogen, and this double bond, which had a hydrogen on it, has instead had an amine group added to it. So when you look at the cytosine, that has an amine group here, this nitrogen here, just a nitrogen, not bonded to anything, and this double bond O over here, what happens is hydrogen bonding. In the hydrogen bonding, you can clearly see this NH2 has these hydrogens, so it's donating a hydrogen towards oxygen, that's getting attracted towards this carbonyl oxygen. This nitrogen has a hydrogen here, that's being attracted towards this sp2 bonded sp2 hybridized nitrogen, which is more electronegative. Okay, and over here again there's a double bond O, so this is also pulling. So you can see that it donates 1 and it takes 2, right? And this guy is donating 2 and taking 1. So C and G, wherever they are, must fit with each other. They must fit with each other. There's no other way for them to fit. Now let's look at adenine and thymine. These are a little harder. If you want, you can pause the video and identify which one is adenine and which one is thymine. Okay, I hope you did that. And you should always remember that adenine over here is a purine, sorry, and guanine is a pyrimidine. Stop, my bad. I said guanine, I meant thymine. Thymine is a pyrimidine. That was a slip of tongue. So here I've drawn this, and it looks a lot uglier because I've tried to include the ribose structure to get it perfect. So when I include the ribose structure over here with a 3' free end and this phosphate group here, it's going to link up to this structure. You can again see the hydrogen bonds over here. This has an NH because this bond had to be broken to give a double bond O. This double bond O, which was participating in cytosine, does not participate here. Why doesn't it participate? Because this double bond O, you remember, is common to all purines. If it's there in thymine, it's also there in uracil and in cytosine. And you can know this is thymine and not uracil, by the way, because of the presence of this methyl group. If the methyl group wasn't here, it would have been uracil, but it's actually thymine. So if you're wondering what happens to this oxygen here and why it's not participating in any kind of hydrogen bonding, that's because there is no corresponding guy here on adenine to bond to it, whereas there is a corresponding guy here on guanine to bond to this double bond O in the case of cytosine. Okay, now you can see that cytosine is taking 2 hydrogen bonds and giving 1, right? Whereas if thymine were to accept another one over here, if thymine were to accept a hydrogen bond over here, right, it can accept it. The only reason it doesn't accept it with adenine is because adenine has nothing to give. If thymine could accept one over here, you might say, look, this is also taking 2 and giving 1. This guy is also taking 2 and giving 1. Can't we just put them together? Like, can't thymine substitute for cytosine? Can't it bond to guanine? And the answer is no, it can't. Because if you look at this bonding here, cytosine takes these 2 from guanine and then gives 1. Whereas thymine has an alternating pattern. Take, give, take. And so, it could never bond to guanine. It has to bond with adenine. So, though it could make this third hydrogen bond, it doesn't. Because if it tries to make it with guanine, it won't bond. And if it tries to make it with adenine, there is nothing for it to bond to. Okay? So, it bonds over here with adenine. Again, you can see adenine has this nitrogen over here. Adenine has this nitrogen over here that is again sp2 hybridized, very electronegative. It will bond to this hydrogen over here and has the NH2 group that will donate this over here. But you know that this nitrogen of the purine group must be bonded to the first carbon, the 1' carbon of the oxygen, of the ribose sugar. So if the 1' carbon is to come over there, then this entire structure must be flipped around. Unless it's at this particular angle, there's no way that this bonding is going to take place. So to ensure that the bonding takes place correctly, this guy must be flipped around. And if this guy is flipped around, then it's going to be the 5' end which is free over here, and the 3' end which is occupied, whereas here the 5' end is occupied and the 3' end is free. Now, this is admittedly this diagram is not properly to scale. I've cut corners with the angles here and there. You can try and work it out for yourself and convince yourself that this is the case. Okay, like here this angle is not correct of this bond, so you might still be trying to envision why do these two need to switch. But if you make it with the correct angles, it's very clear. Here it's a little, due to space constraint, I couldn't draw it out properly. Okay, so now you can see that C and G will always pair. You can see A and T will always pair. You can see that why the 3' and 5' are inverted. I may have told you why thymine can't substitute cytosine, but why can't thymine substitute adenine? Why can't you have two thymines bonding together or two adenines binding together? Remember Chargaff's rule. We formulated it in two ways. We said pyrimidines by purines is equal to 1, or you can phrase it by saying A plus T by C plus G is equal to 1, whichever way you prefer. So if you think of it as saying that the pyrimidines and purines have to be equal in density, that's important. Why is that important? Think of it this way. If I have a purine, okay, sticking out here, it's pretty long. Like it will come almost from this helix to that helix. And on the other side, I'll have a, so one second, let me just change the color scheme here. So if I do it up here, say this is a purine. It's fairly long, and this short green thing at the ending is the pyrimidine. So the purine plus pyrimidine is the length that this comes approximately to 20 angstroms or 2 nanometers. If you had adenine bonding to adenine or thymine bonding to thymine, like a double purine or a double pyrimidine, then there's going to be a problem. Because the enzyme that knits them together, that stitches them together like this, has a certain active site. We've covered what active sites are before. And they have a certain set of dimensions. If it's too thick or too thin in the center, it's going to be hard for it to bond to that enzyme. So it won't get stitched up correctly. Of course, over here this doesn't quite apply. Thymine is fairly small. If it's smaller, a thymine dimer can still form, and it has been seen, but rarely. Okay? Whereas adenine dimers are generally not seen. Thymine dimers are also seen generally because of mutation, but adenine dimers are generally not seen at all because of the fact that they are two purines and they'll be very bulky and it won't fit in the active site at all. Okay, so to summarize, bDNA is a biologically active form. It has 5' and 3' co-occurring at the two ends on opposite strands, and it has base pairing as illustrated here. Thymine will always pair with adenine, cytosine will always pair with guanine. Thymine can take 3 hydrogen bonds, but it won't substitute for cytosine because of the order of the bonds being changed. So it will bond with adenine, which also gives 1 and takes 1 hydrogen bond. Thymine dimers can form, but rarely, because that causes the DNA helical width to shrink. Adenine bond dimers do not form because it will make it too fat. These guys obviously can't dimerize because if cytosine were to dimerize, how could it? It's taking 2 and giving 1. It needs to bond to something that is giving 2 and taking 1. And that concludes our section on the double-stranded bDNA helical model proposed by Watson and Crick. Now we can move on to reading this code and figuring out how to process DNA, and that is the foundation of a subject called molecular biology, and we'll just do the very briefest bit of it. So now we're going to talk about the next thing, which is, as I said, how we read and extract that information from DNA. So Crick, remember Watson and Crick came up with this model? So Crick came up with this idea about how information was supposed to flow. So he thought there was DNA, RNA, and protein, right? These three major things. So we've already spoken about RNA briefly, but he had this realization that proteins are made in little factories called ribosomes. You might remember the term from lower classes. And these ribosomes are in the cytoplasm of a cell. They're not inside the nucleus. The DNA is in the nucleus. And we find RNA lurking around all the time, and it's another nucleic acid. So he thought since RNA is less stable than DNA, what the cell is doing is it takes DNA, it creates an RNA copy of that DNA, of some stretch of the DNA. That RNA copy then gets turned into a protein. Not turned into a protein, the information is extracted from the RNA, which leaves the nucleus and goes to a ribosome. And that information is extracted from the RNA, and the protein is built. So basically the ACGT, the 4-letter code here, becomes ACGU, a 4-letter code here. And this code over here is actually going to tell us a recipe for what amino acids to stitch together to make our final protein. This was his idea. So he named certain processes. He said DNA undergoes DNA replication. So every time a cell divides, you only have so much DNA, but each of the daughter cells should get the DNA, so you double the DNA and give one copy to each daughter cell. That's DNA replication. Then he said that DNA should undergo a process called transcription to create an RNA copy. So what that means is we create an RNA copy alongside the DNA by stitching together little bits of RNA. So those are called ribonucleotides. Okay. So ribonucleotides are used to stitch this together and to create something called RNA. And finally, that undergoes a process called translation in the ribosome to give rise to proteins. You can remember this by remembering the literal meanings of transcription and translation. When someone texts you in Hindi but they're too lazy to use the Devanagari script, so they type in English letters or in Kannada or any other Indian language for that matter, or any language really. When you type in one language but use the letters of another language, that's called transcription. That's a fancy word for it. That's basically what he was saying here. DNA and RNA, they're sort of the same language. It's all A, C, G, only the T and the U are different. And it's a 4-letter language. The main difference is that the bases of the letters are different. The amino acids go from ribose to deoxyribose. It's sort of how there are a lot of Gujarati letters that look like Hindi letters without the line on top. So it's like that. You're just omitting one repeating thing, the O and the deoxy, and then you're changing one or two letters, like you're changing the T to a U and vice versa. So that's why this he called transcription. It's like writing words in your language but in a slightly different script. Then translation is used because proteins are made out of a language of 20 amino acids, 20 to 22, right? And humans have 21, as I said. So we have this translation from the language of nucleic acids to the language of protein that takes place over here. Later on it was discovered that this is true for all cellular life. But there are a few viruses out there that never made the change. Scientists hypothesize today that we used to consist entirely out of RNA. But as cells began to evolve in their modern form, an enzyme called reverse transcriptase, something that does the opposite of transcription, was developed. That reverse transcriptase turned RNA into DNA. And ultimately what happened is all cellular organisms have used DNA as the fundamental thing. So turning RNA to DNA was a bit like backing a memory to a hard drive. That's what it was. So bit by bit this DNA was turned into RNA and it accumulated and it formed the basic genetic code of living organisms. Okay, but there are some viruses around that never decided to make this change. They just have RNA, and so when these viruses infect us, they have another process marked in purple that Crick never thought of. That is called RNA replication, just like DNA replication exists. And from RNA they make more RNA. And the most clever of all viruses is called retroviruses, which include HIV. These aren't viruses that are like listening to retro music, obviously. A retrovirus is a virus that has this elusive enzyme that was present at the beginning of life that converts RNA into DNA. They sneak into us, they're RNA viruses, but they have the genes to make this enzyme. And what they do is, since they want to masquerade as part of us, so we don't realize, hey, you're RNA, let's destroy you because you don't belong here, they turn themselves into DNA. They create a DNA copy of themselves as long as they're inside our cell. And they survive inside our cells in that way. So this is another process called reverse transcription that Crick never foresaw, again shown in the purple arrow. And both of these purple arrow processes are only seen in specific viruses. But the retrovirus strategy is now used everywhere. When we want to create a drug or when we want to create a protein, say we want to create human insulin. Okay, we can't, when a diabetic patient needs insulin, you can't go and take it out of other people. You need to create, because you don't have that much insulin to donate. It's not like blood donation. So we need to donate insulin from some other organism. What we do is, we take our DNA creates an RNA that has the message, that has just the information to make the insulin protein. Before it gets translated into protein, we snatch this RNA out of the cell. Then we put it into a bacterial cell and we infest this bacterial cell with these retroviruses. These retroviruses have this reverse transcriptase enzyme. Along with copying their own RNA into DNA, they'll also end up copying this RNA for our insulin into DNA. And the bacteria will start making our insulin because they'll receive this DNA and they'll think it's their own DNA and they'll start producing it. This is how genetic engineering works. So this is very important. But whatever in purple, Crick never foresaw. So what he did foresee, DNA to DNA, DNA to RNA, RNA to protein, is known by the term central dogma. Central dogma of molecular biology. The central dogma of molecular biology. The thing about this is, see, Crick wasn't a very religious guy, so he didn't know what a dogma really means. A dogma means a certain faith, typically in a religious context, which you accept out of pure belief. There's no questioning involved. Okay? He just thought it meant a central belief. He didn't realize it meant unquestionable. So though it's a scientific thing and obviously it was questioned, it was changed, we added these things in purple arrows as we discovered more things. He gave it this name because he meant it's a central belief. He didn't realize that dogma meant unquestionable. And this got him into a lot of controversy later on for naming it wrongly. Anyways, let's come to DNA replication. Okay, this is the first of the three processes mentioned here. So what happens is DNA replicates via what is called a semi-conservative method. Semi-conservative, which means half conserved. What do I mean by that? Look at this DNA, splitting into two, right? So basically we pull the two strands apart. This is why we have two strands, so we can give one copy to each daughter cell. We split the two strands apart and an enzyme comes and it synthesizes this copy along each of the strands as they come apart. What happens here? Okay, so for that we need to know how to nomenclate each of the things we spoke about. When we were studying this, we spoke about ribose sugar and deoxyribose sugar, right? So when we have deoxyribose sugar and we add just just the nucleic base to it, the nuclear base, it's known as a nucleoside, which means just like we had methyl glucoside and stuff, this is a ribose side, right? We're adding it to the hemiacetylic carbon. So what we're doing is we're forming nucleosides, right? We're using nucleic bases to form ribosides. So it's a nucleoside, or in full form nucleoriboside. When we take this nucleoside, okay, which is basically just our ribose sugar with this nucleic base over here, when we take this nucleoside and we add a phosphate group to it, it becomes a nucleotide with a T, nucleotide. Okay, this is adenosine monophosphate, for example, that's a nucleotide. Guanosine monophosphate, all of these are nucleotides and they are basically the monophosphates, and as we said before, AMP can mean energy source, right? Now there's something else we can do. Do you remember in ATP we had adenine, we had something that I wrote as A, that was adenosine, adenine riboside, okay, linked to a phosphate, linked to a phosphate, linked to a phosphate, right? So that's what basically happens here. We have not just with adenosine, but with any of the nucleic bases we've studied, we can link them to ribose and get a nucleotide. Then we can link them, ribose get a nucleoside, link them to one phosphate group, get a nucleotide, link them to a second, get a nucleoside diphosphate, link them to a third, get a nucleoside triphosphate. This is what happens here. Normally, we use ATP to provide energy for other things. But here, those nucleoside triphosphates like ATP, GTP, CTP, UTP, TTP, all of these will be brought in here. So you have this, say you have ATP over here, which is not yet part of the blue strand, and the orange strand that is coming down over here has a G in it. Okay, it has a G in it. So what happens is the enzyme brings these ATP, GTP, all of these close by, all these triphosphates, and the A and the, sorry, not G, my bad, what am I saying? This is a T over here. This A and this adenine and this thymine, they have this bonding that takes place, the AT bonding that takes place. So this ATP gets sucked in, it gets pulled towards the orange strand because it has base pair, it's formed this hydrogen bonding. So it gets sucked in here, and then what happens is we cut off first this phosphate group and then this phosphate group. That releases energy. That energy is enough for this ribose to polymerize with the phosphate group of the previous ribose on this blue sugar. So what happens is that releases enough energy to stick this adenine. So this adenine became ATP, from that it became ADP, and then finally AMP, which is basically just what we abbreviate as A, right? So it became AMP, adenosine monophosphate. What is adenosine monophosphate? It's adenine then the sugar, and then the phosphate. That's exactly what our A was, right? So it becomes the normal A that we've had in our DNA, and it gets bonded on over here, and therefore it becomes part of the blue strand. And in this way, all of these triphosphates are brought in. We cut off the last two phosphates. Once it happens to hydrogen bond to the opposite guy, we cut off the last two phosphates, generate enough energy to stick it onto the blue thread, and this way we get two copies of the DNA. One goes to each cell after cell division. Perfect. What about creating RNA from DNA? That's called transcription. That's also fairly simple. First, taken a single DNA double strand. One is going from 5' to 3', the other one is going from 3' to 5'. We've already discussed this. So we have an enzyme that comes and opens them up like this. Instead of being intertwined, they become like this. Then so this is the DNA, right? Now we want to create RNA. So we start creating step by step an RNA strand next to this 3' to 5' DNA strand. Okay, this blue box I've done, that's the enzyme. It's moving along the 3' to 5' strand, and it's creating a copy. Let's see what happens. Let's assume over here, let's assume that this sequence is A, T, T, G, C. A, T, T, G, C. Okay, what is going to bond with what? Now, normally thymine, T, bonds with adenine. But here we're making an RNA sequence. Okay, so what will happen is, just like we had ATP, GTP and all these coming up here, over here we'll have UTP coming up. So we'll have a UTP, uracil triphosphate. Okay, this uracil has formed hydrogen bonding with this A, and therefore it comes up with this triphosphate. We cut off these extra two phosphates, we get enough energy, and then with that energy we can start polymerizing. We can start making this long chain of RNA. Okay, the next guy was a T. So this adenine there'll be an ATP that comes up. This A will bond to this T. We'll get rid of the extra two phosphates, and that will provide enough energy to link this onto the polymer. And this way we'll get this long polymeric chain. But what about this guy? This guy and this guy, they were pairing via base pairing, right? So wherever there was an A, there would have been a T here. So we'd have got the letters T, A, C, G. So this is really cool, since this strand has to be complementary to this strand, and since the RNA strand has to be complementary to this 3' to 5' strand, these two strands end up being identical. You can see this is TAACG, this will become UAACG. Obviously we have to substitute U for T whenever we create RNA from DNA. But what's effectively happening is that the RNA strand is identical to the 5' to 3' DNA strand, and it is complementary to the 3' to 5' DNA strand. The reason this happens is because the DNA strands are mutually complementary, and this RNA strand, which is 5' to 3', is complementary to the 3' to 5' DNA strand. So this complement of a complement makes it itself, right? That's basically what we've done. If you want to take it in terms, let's just assume this is a binary code. So instead of 4 digits, just 2 digits, make it simple. So let's say I had 0, 1, 1, 1, 0, something like this as my initial code, okay? And we have two parallel strands, and they're always complementary. It will be 1, 0, 0, 0, 1. Now what I'm saying is, I am creating my RNA code. So when I create my RNA code, I'm bringing in a 1. Okay, so I'm bringing in whatever pairs with 1. What pairs with 1? In this hypothetical example I made out of nowhere, we're saying a 0 pairs with a 1. So this 0 comes and drifts over here. Okay? Then a 1 is going to come and drift over here. A 1 is going to come and drift over here. 1 is going to come drift over here. 0 is going to come and drift over here. These extra phosphates which are over here, so basically the energy source for pushing them into place and linking them up together. So what ends up happening is that this is the same as this in code. Now you might say that's not the case. This TU difference was there. So let's just say for the heck of it that in our RNA, which is the one I've drawn in purple, the zeros have a slash through them. So then you'll say, okay, it looks sort of different, but you can see it's really the same, right? Similarly, when we replace this with this T with a U from this versus this, okay, when replacing T with a U, it's something similar. It's something similar to replacing 0 with a slash 0. The molecule is nearly the same. So basically, the RNA that forms is identical to the 5' to 3' strand of DNA, and it is complementary to the 3' to 5' strand of DNA. And now we can finally study how this RNA directs the making of protein. So with this we come at last to the final major process, the translation. The translation of the information contained in RNA into the information of proteins. How is this going to work? Okay, so this is your mRNA. Okay, let's label that here. mRNA. What does that mean? Well, it turns out Crick wasn't 100% right. Some of the DNA is indeed converted into RNA as a message. Not converted, some of the RNA is created from DNA to function as a message. But there are two other types of RNA that are also created from DNA. When we say mRNA or messenger RNA, we simply refer to the RNA that Crick was thinking about. The RNA that is used to copy a message from the DNA, as in what are the instructions of how to make a protein, and to take it to the ribosome. So I specify mRNA here just to indicate this is the original RNA that Crick had thought about, the RNA that carries our information. This is going from 5' to 3'. This is the length of it. And somewhere along it, there is this particular triplet of letters, AUG. Okay. Now, this is of huge importance in biology, this particular triplet code, because it is what is called the start codon. So, in other words, whatever was before this did not code for anything. Whatever was before AUG did not code for anything. It was just this area that was wasted. It was copied from the DNA because maybe the enzyme found a convenient place to go and bind to it. There are specific places along DNA where the enzyme can go and bind to create the RNA copy. So, this is where it actually went and bound, and this is where the actual message starts. So, AUG signifies a start. So what happens is, you have heard about these ribosome protein factories. What you may not realize is that they typically don't exist the way you draw them. We draw them as these closed structures, but actually ribosomes are made out of two halves, two open structures. These halves come and they fit around the RNA once they find one. Otherwise, they roam around independently. This is called the small half. What is this white thing I've drawn? It's the small half. Now the small half is made out of, actually both the halves. The ribosome is made out of rRNA and some proteins. What is rRNA? rRNA is basically ribosomal RNA. So again, this is copied from the DNA, but it isn't used as a message. It's just part of this factory, okay, that creates proteins. We'll come into what the RNA in here does in a moment. Okay, so this comes and it hooks up wherever with the mRNA, wherever there is an AUG. Now, what the hell is this thing I've drawn over here? This over here is a tRNA. This is the third type of RNA. T here stands for transfer. You're transferring something, okay? So what is this tRNA going to do? It's going to come over here and it's going to bind to this AUG over here. So first, let me give you the process as it happens. I think I've gotten ahead of myself a bit. First, there is this entire long chain. Then this small RNA comes to this rRNA. It comes to the rRNA comes to the 5' end. It moves along until it finds an AUG. Then it gets stuck over there. This is the place where I had labeled it. rRNA plus protein. And this is the small unit of the ribosome. So it has this particular structure. I have told you proteins can be very specific in recognizing something. So it has a particular structure by which it recognizes AUG and it gets stuck onto it. Once this happens, the large subunit will come and get stuck to the small subunit. So the small subunit sticks to the large one over here. So once it finds an AUG and gets stuck to it, then if any large subunit happens to be passing by, it will also get stuck to it. It will be caught by this small subunit. And now the entire ribosome is assembled. So we can start our work. This is a tRNA, a transfer RNA. As you can see, unlike the mRNA, which is straight, and the rRNA, which is entangled in a protein, the tRNA has formed this complicated structure for itself, right? It's come down, then gone in a loop, then gone like this, come back, gone in another loop, come back up here. So it's formed this complicated structure, but what it has basically done is, some enzyme has come and stuck an amino acid onto it. Okay? Now what's going to happen here is, right here at the very base of the tRNA, is what is called an anticodon. Okay, it is a triplet of letters, a triplet that exactly opposite to this AUG. What would that be? UAC. So it's got a UAC over here, so it gets attracted in here and it gets pulled in by the large subunit, right? And then a tRNA which bears a UAC over here has got this identifying tag. So the enzymes which come and add an amino acid here won't add any random amino acid. No. It will add a very specific amino acid called methionine. Do you guys remember that I said methionine is always, it's the amino acid which is the first amino acid towards the N terminal of any protein to be produced? This is why. Because AUG is the one that attracts the ribosome around it. Then this AUG also pulls in this tRNA by bonding to this UAC. Hydrogen bonding, hydrogen bonding, hydrogen bonding. So this tRNA gets sucked into the large ribosome and the large ribosomal subunit, and the large ribosomal subunit kicks it back out again. So the tRNA gets kicked back out. But what it does do is it chops off this methionine when kicking it out, and the methionine gets stuck inside. Cool? So now what happens if we move a little further along in the 3' direction along this mRNA strand? Okay, let's just assume for the heck of it, because there is actually no specification. There is a specification for the first triplet of letters. This triplet of letters, by the way, is known as a codon, and we'll come to this bit that I've written up here on the board shortly. Okay, so the first triplet of letters has to be AUG. We know this because AUG is the set of letters that triggers the small subunit of the ribosome to attract and bind to the large subunit of the ribosome. However, from the second codon onwards, there is no specification as to what it must be. And that's why different proteins are different, because they have different sequences of amino acids after the first one. For the sake of the argument, let us assume that the next codon on this particular chain is, it's the codon triple U. Okay? And then, if this is to be the next codon, then what would the corresponding tRNA look like? Okay. What would the corresponding tRNA look like? So pause for a moment and try and figure out what the anticodon must be. What would the 3 letters be to which this UUU would bond? Assuming that you've got it right, you would have seen that the answer must be A. So a AAA anticodon is the right set of bases at the bottom of a tRNA to bind to a triple U codon. So let's see what's going to be happening here, because it's really deep and it's really interesting. It drives all of our molecular biology processes. The small subunit, having attracted the large subunit, is now more or less just going to be moving along. So we don't need to worry about the small subunit's function anymore. The large subunit is going to bind tRNA after tRNA after tRNA. On occasion, the anticodon of the tRNA will bind. On occasion, the anticodon of the tRNA will bind via hydrogen bonding with the codon of the mRNA. When such a binding takes place, then the tRNA will surrender its amino acid into this large subunit and be chucked out. When such binding does not take place, it will simply be chucked out along with the amino acid. Now obviously, if I have UUU here, then this must be coding for a totally different amino acid. What is that amino acid? That amino acid for UUU is actually nothing but phenylalanine. So phenylalanine, PHE. So now what happens is phenylalanine gets pulled in here, and now we have the chain met-phe. Now you might be wondering, okay, this is going from 5' to 3'. Are these amino acids going to be put together in an N to C terminal direction or a C to N terminal direction? The reason we always write proteins in the N terminal to C terminal direction is that is the way they are produced. So the first codon to be read, which is near the 5' end, it's the first AUG from the 5' end. The first codon to be read always codes for a methylene at the N terminal end. Going onwards towards the 3' position, we start getting more and more codons, and they all code for more and more amino acids till the C terminal position. So let's just keep going like this. Let's say we have some arbitrary codon at some point, some arbitrary codon XXX. Okay, this is our C-terminal amino acid. So it codes, it brings in another tRNA which will have its corresponding anticodon. Let's, I don't know, we could write it as YYI, but who cares? So XXX, corresponding anticodon, corresponding amino acid, and that's the last amino acid. And now we'll end up having a total chain that goes on and on and on from met, phi, etc., 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 until the C-terminal amino acid, whatever that may be. Then, how do we tell the ribosome that you're done with your work? Like, does it keep going till the 3' end, and then does it fall off? Basically, do we have something like this happening? Do we have a scenario like this happening where the ribosomes have come to the very end and then they fall off the mRNA? Generally speaking, no, because the end of the mRNA, just like the beginning of the mRNA, typically has a non-coding region. It has a region where there is no coding being done, like towards the last bit or towards the first bit, there is actually no amino acid that is coded for in this bit. So what instead we do is we have another kind of codon which doesn't code for any amino acid, but which comes after the C-terminal codon and tells it to stop. One example of such a codon is UAG. UAG, UGA, there are a whole bunch of these codons, 3 of them I think. So you have stop codons and they basically pull in another amino acid. Okay, so UAG, what would UAG correspond to? UAG would correspond to AUC. Instead of bringing in another amino acid, this guy would bring something in known as a release factor. Okay, so I'll just write that as RF. For release factor, and this release factor will come again. This tRNA will be pulled into the can be pulled into the large subunit at any point in time, and if this AUC doesn't match with whatever is here, then it will be kicked out. Once it suddenly matches with this UAG, that's a stop codon. It's saying that the protein is over, code no further. Then the binding takes place, and this guy has this release factor cutoff. But unlike the amino acids which are attached to one another, the release factor is a signal. It's a signal to the large subunit that your work is done. Then these two units fall apart, the ribosome decomposes back into its component halves, and we are done with the entire N-terminal to C-terminal polypeptide chain. Okay, and then this is free to fold itself, as we studied, because of hydrogen bonding, into a secondary structure. Then oftentimes it will undergo certain modifications after the secondary structure has been formed. It will be certain new groups will be added to it. When those new groups are added to it in places like the Golgi bodies and whatnot, it will undergo a second layer of folding, the tertiary structure that we studied before. Then various enzymes can come and help form a quaternary structure. So there's a lot of post-translational modification. But this is a process of translation from mRNA to protein. Okay. There are, however, certain features of this code with which we must accustom ourselves before we move on from this point. And I'm going to go over them one by one, and that should broaden your mind to a few of the problems and subtleties and challenges that our cells encounter when they undergo a translational process. Okay. So the features of the codons that we need to study are as follows. The first is a jolly good thing. They are unambiguous. So if there is no mutation in your genetic code, okay, if your genes aren't wrong, your proteins won't be wrong. At least as far as translation, they won't be wrong. As we said, after that they go through a lot of post-translational modifications. That could mess them up. But the proteins produced from the mRNA in the translation stage, these will never go wrong unless the mRNA itself is wrong. AUG is always methionine. UUU is always phenylalanine. And UAG is always a stop factor or a release factor. So that's unambiguity. As I think you can all agree, it's a jolly good thing. It's very important, essential for the way we live. The second one is a horrible term, but it means something very important. I don't approve of the use of the term, but it's used in NCERT, especially if you're a biology student. It might come in your exams, and if you're taking a pure science exam, not an engineering exam, this could easily come even a competitive exam. So I'm teaching you this, but it's not essential in the sense that it's not the right word really. What commaless means here is simply that the amino acids exist without any barriers between them, no commas as it were. Okay, so when I have this AUG and then I have this UUU and then I've gone on to this UAG like this, they don't actually exist in discrete triplets. It's just AUG, UUU, XXX, UAG. It's read monotonically. Why is this important? Why is it important that there are no divisions between them? Let's see what happens with a sort of thought experiment. Let's just assume that this that this U gets deleted. Let's just assume it got deleted. Okay, so then I go AUG is one triplet, and then my ribosome will read the next triplet, which is this UUX. Whatever this X is, it could be a C or a G or whatever. And then the amino acid that gets synthesized, not synthesized, pardon me, but the amino acid which gets added onto the polypeptide chain is going to be a UUC or a UUG or whatever the UUX is, corresponding equivalent. It's going to be the amino acid corresponding to this new triplet. Okay, and then we'll keep on having triplets like this, and then finally, you'll end up messing up the entire polypeptide sequence. You'll even end up messing up the stop codon. And so the ribosomes will actually fall off the mRNA, and it causes all kinds of regulational difficulty. The protein may not even be produced at all, or it may be produced incorrectly. So because there is nothing to say, this is a codon, this is the next codon, and the ribosomes just categorize it on its, the categorization into triplets is done by the ribosome itself. There is no marker on the mRNA that says codons begin here and codons end there. These give rise to the most devastating mutations when one letter is deleted by a mutation or one letter is inserted by a mutation. I think you're all familiar with mutations, X-Men, Hulk, whatever. You get the basic principle. Something enters the body, a chemical or some form of radiation, and it ends up affecting the copying of genes. The genes get miscopied in the stage we studied earlier, DNA replication, and because they're miscopied, they end up creating the wrong RNA when they go through transcription, and then the wrong RNA creates the wrong proteins when they go through translation. But the most devastating mutations of all are not the ones when one nucleic base is replaced with another nucleic base, but where one nucleic base goes missing entirely, because when that happens, every other amino acid in the 3' direction gets screwed up. So it's very dangerous. Similarly, if you insert one, again you're shifting the frame of your reading. Okay, this is what commaless means. So it's an important feature, but a badly named one. Then is the fact that the code is degenerate. So the degenerate and the unambiguous, they're sort of counterparts, and if you've done basic 11th grade maths, you will understand it very simply. Unambiguous means that each and every one of these amino acids has only one each and every one of these codons, pardon me, has only one output. It goes to the ribosomes and only one amino acid is created. So this says that this is 2-1. Okay? So the codon goes to one amino acid. Degenerate says it is from many. So this unambiguous and degenerate is basically saying that translation is a many-one function. In the language of mathematics, this is just saying that translation is a many-one function. So given that it is a many-one function, okay, what does this mean for our genetics? It means, just like I said, that UAG, UGA, and a couple of other amino acids might all have the same function to be a stop codon. Similarly, you might have codons that all code for the same amino acid. Okay, that is fine, because think about it, you have a total of 64 possibilities, right? 4 into 4 into 4, 4 cubed, a total of 64 possible codons, and all these 64 codons actually happen to code for some or the other amino acid, and there are only 20 to 22 amino acids depending upon the species, so obviously there's going to be some redundancy. Many codons can code for one amino acid, but it never happens that a single codon could be coding for 2 amino acids depending upon where they are in the sequence or any such thing. That doesn't happen. So unambiguous but degenerate. Okay, degenerate is from many, unambiguous is 2-1. It's just a many-one function. It's as plain and simple as that. And finally, directional. This I've already explained to you. There's nothing to say. It goes from 5' to 3' and creates a polypeptide going from the N terminal to the C terminal. And that is all you need to know about this machinery. We've covered how nucleic acids behave, and we've covered the 3 steps that we go through the central dogma of molecular biology to extract that information. And now we are done.